So we've already talked about active transport last week. We sort of touched on it. We're going to delve into it a little bit more today. Active transport is the opposite of passive transport. If you remember, things like diffusion is going from an area of high concentration to an area of low concentration. And I use the analogy of like an Axe body spray can, spraying it right in front of me in a big circle. It's not just going to stay there. It's going to diffuse out to the rest of the classroom. People in the front row are going to smell it first, and then eventually, over the course of time, people in the back row smell it. The simple concept of diffusion. Active transport is the opposite of passive transport, because instead of going from an area of high concentration to low concentration, it's like going from an area of low concentration to high. So it's like if you had the ability to individually take those Axe body spray particles that are at the back of the room and all over the room because they've dispersed, and bring them back and like focus them in on that sphere or area that I sprayed and keep them there. If that was entirely possible, that's what active transport would be. So it's going from an area of low concentration to an area of high concentration. So in order to do that, you might need some superpowers or some energy, which is exactly what active transport requires. It requires energy. So that's a little bit of a preamble into what we're going to do today. So for active transport, molecules are moved from regions of low to high concentration. It's kind of like salmon swimming upstream. Makes it a challenge. Now, opposed to diffusion, which can just have things go straight through the plasma membrane of a cell or the outer membrane of the cell, you need some things to do to make active transport work. First of all, you need special carrier proteins that are like embedded in the cell membrane. You may remember from the fluid mosaic model, there are integral proteins that go all the way through from outside to inside. So carrier proteins in the cell membrane bind with the molecule that needs to be transported across the cell membrane. And as I had previously mentioned on our title slide, you need energy. So this carrier protein that binds onto, say it's a food molecule that needs to get, or a nutrient molecule that needs to get inside the cell, it binds to this carrier protein, and then the carrier protein binds onto it, but it also needs energy to sort of morph itself so that if, for example, I have a marker on my hands here, for the people at home who are not seeing what I'm doing, if this marker was the protein that needs to go inside the cell, which is this area, this is, my hands are the carrier protein that are grabbing onto it, but you need energy to morph it so it brings it inside and then gets released inside the cell. So it needs energy to transform the carrier protein. So chemical energy allows the carrier to change shape so the molecule gets transported along to the inside of the membrane. For those of you at home who didn't understand what I was doing there, I'll show you on a diagram on the next slide. And while you're writing that down, I'll also add in the last point, which basically says once that carrier protein has changed shape, the molecule is then released inside of the cell for it to be used up. I'll give you a minute to copy that down. Okay, moving on to the next slide, just making sure everyone's done. We're gonna, as I'd said, get to a diagram. You don't need to copy it down at all. I'm just gonna describe parts of it here. So let's get my tablet up and running. I can't see where this is here. There we go. Okay, so on this we have four different, let's use blue. We have four different things happening here. So we have, first of all, simple diffusion, where you have just this particle right here, and that goes right through, this is the lipid bilayer, also known as the plasma membrane or the fluid mosaic model. So those two layers of those phospholipids that we talked about last week. So this, any simple diffusion molecule will just go right through the cell membrane. So that's like anything like gases or small particles. One thing I do want to direct your attention to is right here. So you have your concentration gradient. Over here you have lots of stuff on the outside of the cell and just a little bit of stuff here on the inside of the cell. So the concentration gradient is going from high to low concentration from the outside of the cell to the inside. When we get into passive transport here, we have two methods. Passive transport can still use both a channel, so you have a little small channel where this yellow particle can go through, but you also have, if you have a bigger molecule like this blue sphere, blue particle here, it'll bind onto this carrier protein, but it's not using energy because it's going from high to low concentration. It's going from area of high concentration to low, so we don't need energy. It binds on to the inside of the carrier protein. You can see that its shape is very similar to that. It's going to have like a lock and key binding. And then eventually the protein will open up to the other side. These sides will separate and release the protein or the particle inside. In the last area right here, we are requiring energy because we're trying to get this block particle or square particle outside of the cell. So we're trying to go from an area that's against the concentration gradient or going up the concentration gradient from low to high concentration. It still has a binding site where you have these square binding site to match this nutrient particle or any kind of particle really. You do need energy to make that protein change shape because this particle can go in here, but we do need the sides to close up, which in turn will also open up the opposite sides. So that requires energy. And then eventually that particle can be on the outside of the cell membrane. So essentially that's what's happening here in this last scenario here. That's what we're talking about for active transport. All right, going to clear my drawings and get us to the next slide. We've talked about it very briefly, endocytosis last week, endocytosis and exocytosis. Endocytosis is the process of bringing something into the cell. So endo is going into, is a way to remember it. So a cell can come in contact with a required substance that's just too large to pass through the cell membrane. So what needs to happen is actually kind of a case of, if you remember from middle school or maybe even earlier grades, playing Red Rover, where you had two teams, one across the field from the other. And I'll show you what I'm talking about here. So let's say you have the blue team who is over here against the yellow team, which is over here. Oh, let's not do those. Let's do circles. So the purpose of this game, if you remember, was to have one team, the yellow team, call over somebody from the blue team. And If they break the chain of people in yellow, then they can go back to their team. If they can't, then they join the yellow team. What sometimes happens is if somebody's running as fast as they can, but they just won't break that plane, what will happen is, let me use my eraser here. We'll take those off and we'll draw in yellow. Someone will run all the way, and if the people are mobile and strong enough, they'll create like this pocket almost, and the person from the blue team will get stuck in there, but it doesn't break. That's kind of what happens for endocytosis. So what happens here is a cell can come into contact with something, say it's a food particle that's too large. So what happens is the cell itself will create a pocket around here, and then it'll engulf that particle and then bring it inside the cell. And then inside the cell, that sort of wall of circles gets broken down, and then that blue X or that particle can then be used up by the cell. So I'll do that in terms of the notes here. The cell can form a pocket or an infolding of the membrane, which then transports the needed substance into the cell. And that's exactly what I talked about with our red rover example there. And as you're finishing that up, there are two types of endocytosis that we're going to get into on the next two slides. The two types of endocytosis are penocytosis or pinocytosis, and the other one is called phagocytosis. Okay, assuming that everybody has that, we're going to move on. I'm going to clear my my drawings because otherwise they'll stay on the screen, and we'll get to penocytosis, also known as pinocytosis. So penocytosis or pinocytosis is the process of engulfing liquid food material into the cell. So it's kind of like, if it's liquid food material, it's like drinking. It's actually, I believe the short form is called cell drinking, is what penocytosis is. So one thing I wanted to mention is that infolding that we talked about, that looks like, you know, it surrounds the particle that's supposed to come inside the cell. Part of the cell membrane that makes like that infolding, that's called a vesicle. So that's called a vesicle. And for penocytosis, the membrane-bound vesicle is called a penocytotic vesicle. These are very small and can only be seen with an electron microscope.
And then hang up again.
Hi, Seth. No, the call didn't stop. Miss Sigurdson just came in, so I'd muted myself.
Oh, sorry.
I'll be back in a sec.
I'm gonna
Okay, we're back. So pinocytosis, we're gonna show, there's a little diagram here at the bottom, or a little picture actually, an electron microscope picture, um, showing how this works. So I'll sort of draw on it as well. Let's try green. Okay, so this is basically the cell membrane right along here. And this is the outside, and this is the inside. This is a lot easier if I look at my computer screen. So what we're looking at here is you have these liquid food particles that are just tiny, tiny little clusters of dots. And they come into contact, and just like with Red Rover, like I mentioned earlier, um, there becomes an infolding or a inbudding or whatever you want to call it. So that's the cell membrane. And again, the particles are all in here as well. And then it starts to sort of pinch at this location here. It starts to pinch and then creates this vesicle that still has those liquid food particles inside. Now, I'd said that it breaks down inside the cell membrane. So in the cytoplasm, there are actually things called lysosomes that do the job of attacking this cell membrane and breaking it down. So then those food particles that are inside can actually be released, and the cell can then use that. The lysosomes will break down that cell membrane, and then other parts of the cell will take the parts of that cell membrane and put them back into the actual big part of the cell membrane protecting the cell. So that's actual pictures of what happens there. I'm going to clear my drawings and get to the next slide, which is not pinocytosis, but it is phagocytosis. So a new subheading there. So phagocytosis, while penocytosis is cell drinking, phagocytosis is like cell eating. So it's got a different name because it forms larger vesicles, and instead of liquid food particles, it's solid food particles. So it often does deal with solid food particles, but it isn't just always dealing with eating. Sometimes it's protective functions. So sometimes it's like a white blood cell protecting us from disease by like reaching out like Spider-Man and going and reaching out to something and then dragging it inside itself so it can break it down. So white blood cells will do that job of like eating, I'm doing air quotes, eating pathogens like bacteria or viruses and coming into contact with them and then breaking them down. So just like I had said there, white blood cells will protect us from disease by phagocytizing bacteria. So while you're writing that down, I'm going to unplug my microphone and plug in some speakers here for the people in class. And hopefully you guys at home will still be able to hear the video. I've got a video of my former roommate in Montreal. He did a psychology degree and then he went to St. Andrews. If you're a golfer, you probably know that St. Andrews is in Scotland, one of the most legendary courses. He took a biology degree there. And after that, he went to Oxford in England to do a master's in biology or a PhD. I can't remember. But anyway, he sent me this video when we did like a FaceTime call essentially back 10 years ago. No, it was Skype. It was Skype 10 years ago. And you'll see an example of a cell, a one-celled organism called a protist, phagocytizing a bacterium or a food particle in much the same way that a cell would do it as well. So just gonna do that, switch things around. Okay, hopefully people at home can still hear me. If not, it's not the end of the world. You're probably sick of hearing me anyway. So I'll show you the video. I'll probably play it twice. And they have British accents.
It's quite a huge bacterium. Oh, that's what you want a video. Oh God. Can you make it?
Insane.
Yeah. Keep adjusting the focus so you can see him do it. Like that.
Fantastic. No, he's so big.
Brilliant, aren't
Oh my God. See all the spines up here?
Yeah.
I didn't know they were so flexible.
they? Yeah, it can totally do full. It would be
Wow.
quite a long bar before he eats anything else, I'd have thought.
That's mental.
I hope it doesn't explode.
That's a full-on a la carte menu he's just eaten in one go.
They can massively do. I think he has almost overreached himself. But you see, he's already making the vacuole smaller.
That's amazing.
And I'd imagine he's much slower with his motion there.
Okay, so you would have seen it reaching out with those little spine things and then engulfing it. And they're quite impressed because it could easily burst. It could actually kill itself by eating too much. I'll play it for you again, and then we'll get on to our last slide.
But he's snared quite a huge bacterium.
Oh,
That's what you want a video.
yes.
Oh gosh. Can you make
That's
it?
insane.
Yeah. Keep
Oh my
adjusting the focus so you can see
God.
him do it. Like that.
Fantastic. No, he's so big.
Brilliant, aren't
Oh my God. See all the spines up here?
Yeah.
I didn't know they were so flexible.
they? Yeah, they can totally do forms. He'll be
Wow.
quite a long while before he eats anything else, I'd have thought.
That's mental.
I hope he doesn't explode.
That's a full-on a la carte menu he's just eaten in one go.
They can massively do it. I think he has almost overreached himself. But you see, he's already making the vacuole smaller.
It's amazing.
And I'd imagine he's much slower with his motion there.
So some people were asking about this this sort of squiggly thing. It's like it's for motility, for movement. It's basically like a tail that whips for movement. So that's sort of what happens in phagocytosis. Now the opposite can also happen. Instead of taking things into the cell, exocytosis is taking things outside of the cell. So exocytosis is exiting the cell. Endo is bringing into, exo is exiting. So it's the reverse process of endocytosis. And instead of taking things in, it's expelling things or getting rid of or secreting materials from the cell. So, question for you here in class. What kind of materials does a cell need to get rid of?
Did you see the rooms outfits for
Like in a good way?
like their outfits they wore?
Oh yeah, sorry, the 90s outfits. Sorry, people at home. Mr. Sloan just came in asking me a question. Yeah, no, those are awesome.
I was looking at, I'm like, that can't be fake. Those bosses that cost that.
Okay, so what might a cell need to get rid of? Amber? Isa? Marissa? Yeah, wastes. Kind of like you, after eating a bunch, at some point afterwards, whether it's half an hour or a couple hours, need to get rid of that food. So cells will also get rid of waste particles or even, waste particles by doing exocytosis. Anything they don't digest, that'll be expelled as well through exocytosis. But just like with endocytosis, it doesn't always deal with eating because it can also deal with protection. We talked about white blood cells. Exocytosis isn't always getting rid of waste. Sometimes, as you learn out in grade 12 biology, the nucleus of the cell has codes like a DNA coding that has instructions basically to make proteins that can then be secreted out of the cell and be used elsewhere in the body. So you can make things like hormones, you can make things like enzymes that just do jobs already all over the rest of the body. So exocytosis, cells can also secrete substances like large protein molecules such as hormones. All right, everybody's finishing up writing those notes. That's the end of the notes for today. I am going to give you, I don't want you to get scared. It says 5-point quiz, but it's not going to be an actual quiz. It's sort of like a wrap up of what we've done for this class. So you don't have to write the questions out. You can just write out your answers if you want. And absolutely, you can refer back to your notes from today if you need help doing that. You're not handing these in. They're not for marks. It's just, again, like a summary of what we did today. So I'll put all of the questions up there, one after the other, and then I'll read them out. And then you can just write them out. And in about 5 minutes, I'll give you the answers. So the first question says, what is the difference between passive and active transport with respect to concentration? Again, you don't need to write down the questions. You can just answer them as you wish. Number 2, what are the 2 types of endocytosis and how are they different? Number 3, what type of proteins help transport particles across the cell membrane during active transport? And number 4, what is the name of the pocket or sphere that's created during endocytosis and exocytosis? So I'll leave those up there for about 5 minutes, and then we'll go over the answers. Give everybody one more minute and then we'll go over the answers. All right, so we're going to go over these and then I'll give you your homework, which you can easily do for people at home, like in the next 20 minutes should be okay, because it's really like 3 to 6 pages, depending on which textbook you have. Okay, so answers. The difference between passive and active transport with respect to concentration. Always think about a concentration gradient, because passive is high to low concentration, while active transport is low to high concentration. So they're opposite. Two types of endocytosis and how are they different are pinocytosis, which is liquid material, and phagocytosis, which is solid particles or solid material. What helps transport particles across the cell membrane during active transport? That is carrier proteins. And lastly, the name of the pocket or sphere that's created during endocytosis is called, and exocytosis is called a vesicle. So typically on a usual 5-point quiz, that's worth marks. This one is not, but on a typical one, I would get you to give your answers to somebody else. They would mark it with a different colored pen and then give it back to you. And it's really just only out of 5 marks. So really just in terms of paying attention to the class that we just did. And usually I'll write it down on the board when we're going to do one of these, so that it's not like spur of the moment. You'll know when you walk into the class, you'll be like, oh, we have a 5-point quiz today. All right, I'm gonna give you your homework. Hopefully people at home, you can see just a black screen because I'm gonna write on it. Was there a number 5? Yeah, the second one was worth 2 points. So while it was out of 5 points, I didn't have 5 questions. Okay, let's do this. Let's do white. So your homework today, I've created a page in OneNote for everybody, and I've distributed to all of your OneNote notebooks. So the page is titled 4-Check Your Progress. And it has active transport in brackets. So like last time, for number 3, you are to read some of the textbook and then answer the questions that are in that page. And again, if you want to type in there or type underneath, somebody mentioned, I think it was messaged me, you can type your answers in there or underneath, or you can also just write out your answers and then upload a picture of that. So if you have the black textbook, so that's a 13th edition with the, what is that? A lizard on the front. You're going to read and it says this in the OneNote page, read pages 77 to 78, and then answer the questions. If you have the white textbook, you're going to read pages 71 to 74. So a few more pages, but I think it's because there's more pictures in there. And then you're going to answer the questions in this page. 4- check your progress, active transport. So it's only 3 questions based on your reading there. And then, yeah, if you are at home, you can log off, and I'm going to stop recording the meeting now.